Hello, my name is Frederick Hurst, and I will be doing a review of the paper Probiotics Mediate Intestinal Microbiome and Microbiota-Derived Metabolites Regulating the Growth and Immunity of Rainbow Trout. I chose this paper because in the coming years, aquaculture production will be more important as we see our wild fish stocks dwindle due to overfishing and environmental issues. The researchers decided to do this paper because they wanted to study more about the mechanisms behind feeding fish probiotics. Because most papers leading up to this paper only study the what probiotics do to fish, and not so much the mechanisms, the how and why. So researchers really wanted to do a deep dive on this topic. They did this by taking two probiotics. One was Bacillus valencina, and the other one was Lactobacillus sakei, and feeding them to fish, and then afterwards studying their growth, their immunity, their serum enzyme activity, and their resistance to the pathogen Aeromonas salmonitica. I find this research area important because the demand for animal protein is going to drastically increase in the next 20 to 25 years. This is mostly due to developing nations, which, as they continue to get wealthier, will have a higher demand for quality protein. And this has been shown in societies across the world, that as wealth increases, people are willing to spend more on protein. It's really interesting phenomenon, but it's definitely been seen in many societies, and it's being seen in some countries in South Asia now. In order to meet this demand, however, production in protein will have to increase, and it will also have to increase sustainably, because we cannot continue to take so much from our systems and keep putting them into this unsustainable practices, because we won't be able to meet this demand. So in order to meet demand of protein demand in the next coming years, aquaculture is going to have to drastically increase. And this is especially true because, as you can see in this graph, that wild capture fisheries are going to decrease. And this is because we've overfished our stocks and all the environmental pollution we've caused, which makes the fish populations unhealthy. So we've decimated them. And in order to make up for this, we're going to have to use aquaculture to grow our fish. Now, there are two main types of aquaculture farms, and you can see them here. We have, on the left, a land-based farm, and on the right, an ocean-based farm. Now, both farms have issues, uh, one of them being overcrowding. When you put these fish all together in a tight area, you start to see a lot of issues such as disease, and too much excrement goes into these areas. And we also have the problem that these environments are all monocultures usually. You're usually growing one type of fish, not several. Um, in the ocean, you'd have things such as oysters, which clean the environment. You'd have different fish which have different actions, like bottom feeders, and you'd have other ways. You'd have plant life, but these are very, very simple You just make a pen, you put the fish in them, and you feed them. And one of the problems with these systems, coming back to this chart here, is these farms are actually all fed on wild fish that are caught. And this makes it imperative that we get the most out of what we can on these aquaculture farms, because we're feeding them from wild stocks that are we feed them smaller fish, and we're incredibly limited to what we can produce due to the fact that we feed our fish these wild fish stocks. So it's important to be able to have marketable product, like every fish is marketable. And this doesn't always happen because we overcrowd the pens. As you could see, these pens hold thousands of fish. And when you overcrowd them like this, you start to see some diseases. And these diseases ruin the fish's flesh and make them unmarketable. The bottom picture here is a picture of rainbow trout, which is actually what the authors of this paper experimented on. But the researchers in this paper are looking at a particular disease, Aeromonas seminitica, and it causes ulcers and boils just like these two pictures show, and it can really make us lose a lot of marketable fish. So we have to try and figure out ways to not only cause the fish to grow, but make sure they don't suffer fatalities or even just become unmarketable due to these diseases. So the deep the goal of the paper is to really do a deep dive on the mechanisms that probiotics utilize to improve fish health. And this is different from most studies, as I said before, because most of them just look at the results of the probiotics in the fish they're studying rather than what, why, and how the probiotics change the fish to cause the results that we're getting. And I think understanding this nitty-gritty, this really how do the fish interact with the probiotics and why and what they're doing is important, because maybe there's a shortcut we can take, or maybe we can use this to develop some other medicines, or there's always ways to use something besides the surface level. So it's always important to understand the why, what is happening. So here is some background from previous studies that looked at probiotics in fish health. So many studies find that probiotics can help lower fish mortality. However, we're unsure of why. Another study known as titled The Identification and Characterization of Lactic Acid Bacteria Isolated from Rainbow Trout with Inhibitory Activity Against Lactococcus cerevisiae found that Lactococcus and Leuconostox are two effective probiotics that can prevent a disease from the bacteria Lactococcus cerevisiae. And this is kind of similar to the basis of what the authors are looking at. But the authors of the paper I'm presenting really wanted to understand how the fish react to the probiotics, not so much, not, it wasn't always only focused on how disease is prevented. This paper by T. Perez et al. Also investigated how probiotics could survive ingestion and make it to the gut, because this is incredibly essential for probiotics. They can't do anything unless they actually get to your gut and replicate and then start working, okay? If you just pass them along or if they don't make it, they're no good. So coming to the first graph, figure 1, we can see that the authors looked at the surface first, at the effects of the probiotics on the weight and fatality and the growth of the rainbow trout. So here we can see at the bottom left, we have the control, which weighed about 50 to 52 grams, compared to the BV and LS group, the BV being the Bacillus valenciensis and the LS being the Lactobacillus sakei. And we can see this Bacillus valenciensis is incredibly good at increasing the weight gain for these fish, which is the first benefit we can see from having probiotics. And we can see the same in the lactobacillus as well. We also can see that the growth rate is accelerated compared to the control in the lactobacillus and the bacillus probiotics. Now, this is important because obviously in aquaculture, you would want fish to grow faster and you'd want them to put on more body weight faster. So this looks promising. But in this third graph, we can see that of the three groups, when presented with Aeromonas salmonitica, only the Bacillus valenciensis is able to keep a low fatality rate compared to the other two groups, which is interesting because I think it really shows that the even if you see positive results in these first two in body weight gain and growth rate, it does not mean you will see improvement in fatality rate. And this is going to come back later in the paper because it's mostly based on the researchers believe that there is a correlation between what these probiotics are upregulating and the genes they're activating and how they're stimulating the immune system. But for now, we're going to look at this figure, which is intestine morphology and treated versus the control. And this is a image of the villus. Now, villi are important because they allow organisms to absorb nutrients as well as many other things. And it's important for the health of an organism because we see in celiac patients, they have completely flattened villi. And this means they're not able to absorb nutrients. And then we start to see the characteristic issues associated with celiac disease, which, if left untreated, can lead to someone being incredibly sick and unhealthy and malnourished. So it's important to keep your villi healthy. And it seems that the Bacillus valenciensis is pretty good at doing that. I think figure C is very interesting in that it looks very different from A and B. And here we can see the actual measurements. And we can see that the probiotics, at least the bacillus group, increase the villus height and the villus width and the thickness, all three, which leads to a healthier gut as we can see here. Now, the next part of the paper starts to talk about how after being exposed to these probiotics, the bacteria, the fish, are start to have differentially regulated enzymes. And we start to see increase in activities of a lot of antioxidants, which can lead to less inflammation and is associated with less disease or irritation. So less inflammation, better health, and it's all caused by these antioxidant enzymes such as superoxide dismutase and acid phosphatase. And we can see that the probiotics have a significant difference from the control in upregulating these enzymes. So by being exposed to these beneficial bacteria, the enzymic activity in the gut and intestine can really change. So besides this, researchers found that the compounds in the gut also change. And this is mostly due to populations of the microbiome changing. So we can see in these graphs that the relative abundance of different species depends on the probiotic administered, because all these fish are fed the same diet. So the significant change comes from the probiotics that are administered with the diet. So we can see that there's some interaction between the bacteria that live in the microbiome based on what we're adding. So, for example, the lactobacillus increase the the relative abundance of firmicutes, whereas the bacillus seem to increase the amount of proteobacteria in the gut. And researchers talked about in this article that a lot of these different species produce different compounds, such as malic acids and different, and they break down different compounds too. And this, once again, usually helps prevent inflammation. But the other thing that all these different probiotics and bacteria do is they also affect the immune system, but we'll get more into that later. So we can see that there's a lot of different changes in what's produced based on these cake charts. And this is due to some differential genes being activated, but also due to the diverse populations of microbes. So, for example, we can see that there's differences in lysine degradation and tryptophan metabolism and carbon metabolism. There's so many differences, and it's all because we started to change the microbiome of these fish, and we start to see them get healthier because of all these downstream effects. So the next part that the researchers studied was this web. They create this web based on the microflora, the metabolites, and the genes that are expressed. So The when you add, say, Xanthomonas, or we can study, we add Bacillus valenciensis, and let's say it increases the Xanthomonas population because let's say, one creates a compound the other likes. This can lead to an increase of sucrose. And this starts to change some of the genes that are expressed in the fish too. And by increasing some of these, we also see different effects on the fish. So by producing more L-malate, we start to see some upregulations of this gene like IGFBP2, and some of these are linked with antioxidants. Some of these are linked with the production of antioxidants, and some of these are linked with the innate immune system. So we start to see genes related to the innate immune system coming online, and they get upregulated, and they kind of it puts the body more on guard. And this is why we see the resistance to the Aeromonas semimitica, because the body starts to, the fish's body starts to get more hyper like the immune system is on high alert, and this allows it to fight off infection easier because it's on high alert, but it's not attacking itself in a bad way where you would see with inflammation. So moving on to the methodology of this paper, the authors use several methods, but to cover a couple, for the metabolomics, they used ultra-high purity liquid chromatography to analyze the intestine samples, and they coupled that with mass spec to identify the compounds. To study the intestinal microbiome, the DNA went through several preparation steps, and then it was sequenced on a Nova 6000 platform, and then several different types of software were used, such as Trimomatic, Muscle, and Chime. For transcriptome anomics, after they extracted the RNA using gel electrophoresis, the researchers used an Agilent bioanalyzer, and then the samples were put onto an Illumina platform for sequencing, much like we learned in class. It's worth noting that the diets were kept consistent for all the fish, but 6 were randomly selected at different points in time at 10, 20, and 30 days. As far as statistical methods go, researchers used several, but some worth noting are the Pearson correlation analysis, which was used for the correlation of the microbiota. And then they also used a one-way analysis of variance and a Duncan's multiple comparison test for the growth enzyme activity and gene expression parts of the experiment. So the results of the article show conclusively that probiotics can cause many morphological changes in fish, and this will range from increased disease resistance to changes in serum enzyme activity, increased growth, and changes in intestinal metabolites. However, I would say that much research still needs to be done to see how these probiotics can help protect against other organisms. I also think research is needed to see what other potential probiotics are out there that can help fish fight these diseases. And in conclusion, probiotics can affect the fish health in so many different ways, and it all probably depends on the strain and type of probiotic that the fish is given. These probiotics are important because they stimulate the innate immune system. They can provide increased resistance to pathogens if it's under the right circumstances. They upregulate certain genes, and they increase weight gain. The probiotics in the microbiome can also change some of the compounds which are produced in the fish's gut, which can help make anti-inflammatories and antioxidants. And finally, I have my questions for you guys to think about. So I wonder, do you think probiotics present a better option for fish health versus the more traditional methods such as antibiotics and vaccines? How do you think the local environment affects a fish's microbiome and health? Do you think that using probiotics to help fish will allow for farmers to continue to overcrowd farms because of the ability to resist disease? And do you think this will lead to more health and environmental issues? How do you think the upregulations of some genes and the downregulations of others help bacteria create a mutualistic relationship with the organisms they inhabit? And finally, how would you design a follow-up to this study? Here are my references, and thank you for listening. Have a great week.